I'm Marissa Vergara, and my protein is on actin. My scientific journal is from the Journal of Biological Chemistry, and it's titled Actin, Protein Structure and Filament Dynamics. It is written by Marie-France Carlier, and her research was done in France in 1991. So a lot of key aspects of her research paper is the structure of actin, ATP and divalent cation binding to actin, and mechanism of polymerization and involvement of ATP hydrolysis. In my paper, I will be discussing the history of actin, the discovery of it, and how it was discovered, and just give back information of actin. That way, it gives kind of a foundation of knowing what actin is. I will also talk about the structure and the function of actin and why it's important for a cell. I will also talk about where actin can be found in the types of cells or in the body as well, or if it's in other species. And I will also talk about the proteins bounded to actin and why they're bounded to actin. So for the scientific journal, how that comes into play, I will discuss why actin is tied to the scientific journal and why she used actin as a key topic. In her paper, she does discuss the ATP role that plays with actin, and also the polymerization and how the ATP hydrolysis comes into play as well. And I will discuss that in my paper. In her paper, she also discusses how actin has been, how it has advanced some researches and how it could possibly advance some researches. And I will also tie that into my paper as well, as far as how actin has impacted the medical world and the chemist world as well, because of the actin and what it's made of. So I'll be discussing those things in my paper, and I hope that a lot of her paper will help in mine as well, as far as discussing what actin is and how actin is a key part of life and cells.